when we first had the idea for the symposium, one of my ideas was that this connection of how we're treating patients, hearing from the patients, and then research that's being done would be a really nice way to blend things together for this community. And it's been really, really rewarding to watch how that's evolved over the years, and people like Kate who are willing to get up and share their story. So thank you again for that. We're going to move on to a series of talks, which are this morning loosely related to leukemia and immunotherapy, and then this afternoon we'll have a session more for solid tumors. The number of abstracts we received this year was an all-time high. I forget the exact number, but it was, thank you, Lauren, 70 abstracts for those who didn't hear. And so then it made it really challenging to pick those that we would like to have presented orally. We try to have a mix of science that's being done at the different institutions that CCF supports, as well as from different people at different levels of training. So I would now like to invite James Kronk from the National Cancer Institute to present his talk entitled B7H3 Nanobody CAR-T and Soluble TREM2 Genetically Engineered Myeloid Cell Therapy in Murine Synergistic Glioblastoma. And I don't think I could have done that with one breath, but I'm going to trust you to explain that all to us now.
I'll do my best. All right. So first of all, I just want to say thank you to the organizers for giving us the opportunity to present our work thus far on this project, and of course CCF for funding this project last year. And I'll present to you today our progress thus far. So again, as was just said, today I'll be talking about combination therapy with B7H3 nanobody CAR T cells and soluble TREM2 genetically engineered myeloid cell therapies in murine syngeneic glioblastoma, but I'll also present some data in other solid tumors as well. All right, so framing the problem, immunotherapy for solid tumors. This is a promising approach that really requires more refinement. So the current standard of care for most solid tumors is going to include a combination of surgery, radiation, and chemotherapy. And while immunotherapies such as CAR T cells are now becoming a standard therapeutic option for relapsed refractory leukemias, unfortunately in the context of solid tumors, immunotherapies really are mostly having partial or even no response for solid tumors. So, more success in leukemias than for solid tumors. And so our project is really geared towards trying to tackle this problem. To discuss our project specifically, my co-PI on this project is Dr. Anandani Nelan. She's a pediatric neuro-oncologist at NIH, where I also perform research in the laboratory of Rosie Kaplan. Glioblastoma is an aggressive brain tumor. It has very high mortality rates and limited treatment options. So most patients with glioblastoma survive less than a year post-diagnosis, and depending on the study you look at, something around 5% may be alive 5 years post-diagnosis. So it's a dismal outcome, unfortunately. The incidence for this cancer starts to increase with adolescence, but it does affect patients of all ages, just increasing with age. Now, to try to tackle this problem, we've developed a fully murine or mouse version of a B7H3 nanobody CAR T cell to target solid tumors in fully immunocompetent syngeneic mouse models of cancer. Now, this is an important point because a lot of immunotherapies are studied in immunocompromised mouse models because we're using human cells and human cancers in mice, and if they had a full immune system, the mice would just clear the tumors. And we really feel that if you're going to be studying immunotherapies, you need a model that has a normal immune system because your immunotherapy is going to have interactions with the immune system that you just can't model without a normal immune system. Now, the model we're using is a fully immunocompetent glioblastoma model that was developed at the Center for Advanced Preclinical Research, or CAPR, at NCI Frederick. This was specifically designed to mimic human glioblastoma with specific mutations seen in that cancer. And I just want to note that this is a very aggressive model. So they've attempted many ways to try to treat this cancer in mice, and thus far nothing has resulted in cures, maybe just lifespan extension. So we're intentionally kind of stacking the deck against ourselves because in reality for our patients, the deck is stacked against us. So we think that's the right approach to take. So in terms of the target that we've picked for our CAR T cell to target glioblastoma, B7H3, B7H3 increases with tumor grading. So as you go up to grade 4 or glioblastoma, you get increasing B7H3 expression. When you compare to other subtypes of gliomas, B7H3 is specifically increased in glioblastoma. And of note, B7H3 expression really correlates with decreased patient survival in both low and high grade gliomas. So both from a prognostic value and expression of the protein, B7H3 seems to be a good target for glioblastoma. So this is a good potential CAR T cell target. The next question is, do our mouse models actually express B7H3? And we've interrogated several mouse models that we typically work with. Of course, we looked at our glioblastoma model that we're working with up at CAPER, and it does, in fact, express B7H3 at nice high levels by flow cytometry and immunofluorescence. And I'm also showing you here an osteosarcoma model that we often work with, and I'll be showing some data with that later, which also expresses B7H3. Now, we took our B7H3 mirroring CAR T cells and tested in a dish whether or not our CAR T cells are effective at killing these tumors, and they are. So when we compare against an antigen-irrelevant CAR T cell, so CD19, normally used to target leukemias, compared to our B7H3 CAR T cell, we get very effective tumor clearance in both our osteosarcoma and glioblastoma models, and concomitantly, we also get nice high levels of IL-2 and interferon gamma expression, as you would expect with CAR T cell activation. So our CAR T cells work in a dish. Let's put them into a mouse and see what happens. And we've just now started doing these in vivo experiments, but we're pleased with the results we're getting thus far, and we think it's showing good promise that this therapy may have some legs. So just to go through how we do these experiments, we stereotactically inject glioblastoma cells into the cerebral cortex of mice, allow them to grow for a week. We precondition the mice with cyclophosphamide fludarabine chemotherapy. This is standard preconditioning for CAR T cells, both in mice and in humans. And now I'm going to summarize like probably 6 months plus of work by our collaborators up at CAPER to get this up and running in just one sentence, where we have this permanently implantable catheter system that we put into the lateral ventricle, or the fluid-filled space of the brain of the mouse, so we can do multiple serial injections of CAR T cells. This mimics more so what we're doing in patients nowadays, where we put in Ommaya reservoirs and instill CAR T cells directly into the ventricles of the brain. So we give 2 million transduced CAR T cells weekly for 3 weekly doses, and we're monitoring tumor volumes by MRI weekly. So in our first experiments here, we were pleased to see that we got up to 20% cures in our B7H3 CAR treated animals versus our antigen-irrelevant CAR, CD19 CAR treated animals. And while in these experiments it wasn't quite statistically significant, we weren't sure what effect size we were going to see, and in a post-hoc power analysis, we think that we need to just basically double our group sizes, and hopefully in future experiments we'll be able to solidify those statistics. And just to show you what our MRIs look like in these animals, you can see that over the course of about a month to a month and a half, our animals that haven't received any treatment grow very large tumors, as do our CD19 CAR T cell treated animals. And I'm showing you 2 different examples of animals treated with B7H3 CAR. On the bottom is an animal that was cured by our CAR, and then on the top row is an animal that was not cured, since we do get a mixed response with that treatment. So our B7H3 nanobody CAR T cells are showing promise against our GBM model, but we're seeing an incomplete curative response. And this is actually consistent with the CAR T experience in other solid tumors. So the question now is, what prevents CAR T cells from completely clearing solid tumors? And one of the big problems that we think we're facing here is an immunosuppressive myeloid cell response that's happening in tumors. So most solid tumors are full of immunosuppressive myeloid cells, macrophages, myeloid-derived suppressor cells, or MDSCs. And these are the cells that are expressing some of the things that we commonly clinically target when we talk about like checkpoint blockade. We're really targeting these interactions, PD-1, PD-L1, which is on tumor-associated macrophages, CTLA-4 and CD80-86 interactions, right? But one target that has become more recently of interest in the literature is triggering receptor expressed on myeloid cells 2, or TREM2, which is an important regulator of myeloid immunosuppression. So while TREM2 is expressed in normal physiology, notably in microglia in the brain, which I'll talk about a little bit later, one of the places where it's really seen to be expressed at high levels is in cancer. It's been seen in most solid tumors that have been interrogated. We've found high expression of TREM2 on the tumor-associated macrophages. So what is TREM2? Well, I'm showing you here a complicated signaling pathway diagram because it is a complicated signaling pathway. It's not involved in just one pathway. It affects a lot of different processes in the cell. It's a scavenger receptor with many ligands. It's involved in phagocytosis. It's broadly expressed on TAMs, as I said, and it's mediating pro-tumorigenic and immunosuppressive functions. But most importantly, previously published work has demonstrated that TREM2 loss of function or inhibition increases macrophage inflammatory responses and sensitizes tumors to checkpoint blockade. So there's good rationale for targeting TREM2 in cancer, and this is some of that previously published data. So if you look at TREM2 knockout animals as compared to wild type in a mouse model of sarcoma, you get decreased tumor growth. This is mediated actually through adaptive immunity. So if you deplete CD8 T cells, as seen in the pink line here, as compared to TREM2 knockout animals alone, you lose that treatment effect. When you combine TREM2 knockout with checkpoint blockade, you get an enhancement of the TREM2 knockout effect with some cures. And you get a similar response when you do TREM2 inhibitory antibody with checkpoint blockade. So doing dual targeting with TREM2 and something to enhance the adaptive immune response is synergistic and seems to be a logical targeting strategy. So we decided to take our previously developed genetically engineered myeloid cell platform and make a genetically engineered myeloid cell that makes soluble TREM2, with the idea that soluble TREM2 is going to act as a decoy receptor to block TREM2 signaling, relieve immunosuppression, and allow for better adaptive immune responses against the tumor. So of note, just real quick, soluble TREM2 is actually an endogenous cleavage product of TREM2. TREM2 is a transmembrane receptor which is naturally cleaved, so all of us have soluble TREM2 in our body, so this is not a foreign protein. And this is just an infographic kind of explaining what we hope is going to happen with our therapy. So we have our, in the normal situation, we have our tumor with suppressive macrophages expressing TREM2 and TREM2 ligands that suppress tumor-reactive lymphocytes that would otherwise attack the tumor. And when we add in soluble TREM2 gemmies, what we envision is that they go into the tumor, they secrete soluble TREM2, block that TREM2 ligand receptor interaction, and release tumor-reactive lymphocytes to then respond to the tumor. So first thing, we had to make our gemmies, which we've successfully done. In brief, to make gemmies, we isolate murine hematopoietic stem and progenitor cells, culture them with a cocktail of growth factors and cytokines to make them into myeloid cells, and transduce them with a lentivirus containing the soluble TREM2 gene. These cells express nice high levels of soluble TREM2, and just to show you what these cells actually look like by flow cytometry, they're almost all CD11B positive, so depending on the prep, they're 80 to 90% CD11B, so myeloid. Some fraction are going to be Ly6C positive, so monocytic. Some fraction are F480 positive, so monocytic macrophage lineage. And of note, none of them are ever Ly6G or CD11C positive, so not granulocytes, not dendritic cells. So we have these myeloid monocytic progenitors. We know from our prior work that they will track to solid tumors and deliver our therapeutic protein product. So we now have the therapy. What are the best models to test it in? So the answer is going to be a tumor model that induces TREM2 positive macrophages. So to decide on tumor models, we did an in vitro screen where we develop or make bone marrow derived macrophages and then take tumor conditioned media from various tumor cell lines, skew the macrophages in a dish for a day, and then analyze by flow cytometry. And what we find is that as compared to unskewed macrophages down here, all of the tumor lines we tested, so 2 glioblastoma lines, a rhabdomyosarcoma and osteosarcoma line, all induce this TREM2 positive, PD-L1 positive immunosuppressive TAM phenotype, quantified over here. So we had plenty of tumor models to potentially choose from. So we decided to go ahead and start actually originally in this project with the F42010 murine syngeneic osteosarcoma line. And this was because we saw abundant TREM2 positive macrophages in these tumors in vivo. So by 2 markers of macrophages, IBA1 and F480, we see abundant macrophages in the tumor. And by flow cytometry, we can see that they are very high expressors of TREM2. So we have our TREM2 macrophages present in the tumors, so logical to target TREM2 in the model. So we went, we're utilizing our standard GEMI treatment paradigm for this, where we inoculate with a tumor, we wait 10 days for the tumors to grow, give cyclophosphamide, fludarabine, preparative chemo, just like CAR T cells. Just like with CAR T cell therapy, we find GEMIs greatly benefit from this preparative chemo, which is an active area of investigation I won't be talking about today as to why that happens. 2 days later, we give the GEMIs, and then we monitor survival and tumor growth. And we were pleased to see that we got reduced tumor growth with our soluble TREM2 therapy, and we got a moderate lifespan extension with the Cy/Flu PrEP plus soluble TREM2 Gemmies, which was in line with what's been previously seen with TREM2 targeting therapies. So in sum, for this portion, we successfully created our Gemmies. We took a tumor model, an osteosarcoma tumor model that contains abundant TREM2 positive macrophages, and when we treat it with our Cy/Flu preconditioning plus soluble TREM2 Gemmies, we got reduced tumor growth and prolonged survival, which is in line with what is seen with TREM2 inhibition or knockout, so biologically made sense to us. So this data supports the hypothesis that our soluble TREM2 Gemmies may enhance immunotherapies like CAR T cells or checkpoint blockade by relieving immunosuppression. So at this point, we decided we wanted to combine these Gemmies with our B7-H3 nanobody CAR T cells in our murine glioblastoma model. So just to take a step back, why are we doing this in brain tumors specifically? Because when we envisioned this therapy, we actually thought brain tumors would probably be the ideal model for it. And the reason is because most brain tumors are heavily infiltrated by immunosuppressive microglia and macrophages, so we have two cell populations to target here. TREM2 is highly expressed by both microglia and tumor macrophages, so in a normal brain, microglia are already expressing TREM2, so we have an additional cell population to target. In addition, while TREM2 blocking antibodies have been demonstrated in preclinical models and peripheral tumor models, we don't anticipate these are going to work in the brain because of the blood-brain barrier. In fact, cell therapies are better able to traffic into the brain than antibodies are able to get into the brain. There's also data from the Alzheimer's literature that soluble TREM2 on its own can induce inflammatory responses in microglia, as seen down here. So we're hoping that we can actually induce just a de novo inflammatory response with our therapy. And there's also more recently been published TREM2 knockout or knockdown in a different glioblastoma model in mice. Prolonged survival and can enhance checkpoint blockade. And while this was non-curative in this model, they did get lifespan extension, so again, supports the idea that TREM2 targeting is a logical strategy for this type of cancer. And while we haven't, we're actively right now doing our combination therapy studies. I just wanted to show you some of our in vitro data looking at what our glioblastoma line that we're working with does to hematopoietic stem and progenitor cells in vitro. So we get, when we take hematopoietic stem and progenitor cells, so these are agnostic hematopoietic stem cells, and co-culture them with our glioblastoma tumor cells, wait 6 days, and then analyze them by flow cytometry. When we look at the immune cells that are left, it turns out that our glioblastoma basically turns almost all of those hematopoietic stem cells into macrophages, so CD11B positive, F480 positive macrophages. And essentially, all of those macrophages are TREM2 high and express PD-L1. So this tumor is very good at making TREM2 positive macrophages. So we're hoping that we're going to see nice efficacy with our combination therapy in this model. So just in summary, graphical depiction of what we hope is going to happen in our treatment. We have our no treatment condition, where we have suppressive macrophages with TREM2 signaling, suppressing lymphocyte responses. When in the context of CAR T cell therapy alone, what we understand likely happens is we have our immunosuppressive macrophages blocking CAR T cells and endogenous lymphocytes, and you get a little bit of a tumor response, but because of that suppression, you don't get complete tumor clearance. With our, as we discussed before, with our Gemmy therapy, what we expect to happen is we relieve immunosuppression by blocking TREM2 signaling, and we get a little bit of tumor clearance, but it's not enough to cure. So what we're hoping is when we combine these therapies together, we're going to relieve immunosuppression through the soluble TREM2 Gemmies, and then we have our active therapy that is now released to completely clear the tumor because we've relieved that immunosuppression. So in conclusion, so far we've shown that our B7-H3 nanobody CAR T cells are showing promise in our syngeneic glioblastoma model. And as I mentioned, we're going to be increasing our Ns for our cohorts to achieve statistical significance on that. We've shown that our soluble TREM2 Gemmies with Cy/Flu preconditioning reduce tumor growth and prolong survival in our osteosarcoma model, which is biologically logical because it's fitting with what's seen with TREM2 inhibition and knockout in previously published data. So moving forward, and what we're actively doing right now is combining our CAR T cells with our soluble TREM2 Gemmies in our glioblastoma model. And as I said before, we're anticipating or hoping that we're going to see a synergistic effect with these two therapies. And to note, we're going to be sticking with the current CAR T cell dose because actually we want a subtherapeutic treatment because we need some room to improve with our combination therapy. And since we know that our soluble TREM2 Gemmies have some efficacy in our osteosarcoma model, we're going to go ahead and also do this combination therapy of B7-H3 CAR and soluble TREM2 Gemmies in osteosarcoma as well. We'd love to be able to impact as many patients as we can. There's good logic behind doing therapy in both. B7-H3 is also expressed in this tumor model, so we will be pursuing those studies as well. And I just want to, of course, thank everybody in the Kaplan Lab, Rosie Kaplan, Sabina Kachanowska, Anna Nellen, my co-PI on this project, the whole lab that developed the B12 binder for our CAR T-cell, and the Euler lab that performed all of the in vivo GBM studies, and of course CCF for funding this project.
It's amazing that we're still working so hard on brain tumor therapies this many years into knowing that they're difficult to treat, but yet we're continuing to, I think, make some progress. We're getting very good at potentially curing these in mice. So the question I have for you is, as you think about translation into humans, how much of this has to be completely rebuilt of your models in order to start going into human therapies? Because I would imagine, just like with the mice, you're going to have to build these things in layers. So where do you have to start from to start to say translating this into human glioblastoma?
That's a great point, and we think about this a lot. So, for instance, our soluble TREM2 Gemmy, we didn't get into all the details today, but for instance, that's already designed as the human version of soluble TREM2 for faster translation. We know from work of others that the human version of soluble TREM2 is just as functional as the murine version, as an example. We're in the background. Dr. Nellen is also working on this B7-H3 CAR, but other B7-H3 CARs as well, trying to evaluate which CARs are going to be most effective in patients, right, as compared to mice, because we have our syngeneic mouse CAR, right, which is what we need to work in animals, but it isn't necessarily going to be the best candidate for humans, right? So we may, for instance, decide that we're going to use a CAR that seems to be a better candidate in humans for a combination therapy in a future clinical trial, right?
Yeah, other questions? Yes, Julie, right here. And then after that, raise your hand in the back so we'll catch you. Okay, Steve, you want to catch? Okay.
Thank you. That was a really fantastic talk. I have a question that maybe I missed and you presented, or maybe you haven't done yet, which is in those glioblastoma models where the tumors did not respond to the B7-H3 nanobody. Have you studied those tumors? Do you know what cells are in those tumors?
We have the tissues, and we're actively working on analyzing and staining those. So, yeah, I don't have that data yet, but yes, we're very interested in that, of course, right? Yeah.
Nice talk, Jim. I have two questions. First, I think I missed this, but the Gemmies, do you put them right into the brain, or do you do peripherally?
Yeah, so we actually we have a system where we put a permanently implanted catheter. So the mice actually have an implanted reservoir under their skin in their back with a catheter that runs up into the lateral ventricle, and we're actually able to instill serial infusions, as many as we want, right? Because it's actually not a surgery each time. It's like an Ommaya reservoir, basically, right? And so we're actually putting it right into the CSF in the lateral ventricle. Yeah.
And my second question is, have you thought about doing it stepwise, doing the CAR or the Gemmy first, releasing your myeloid cells, and then adding CAR T for the attack, or are you going to do it together?
Absolutely. There's another group I'm working with on a PDX model, so a human brain tumor model, where we are investigating that question, and we do think timing is important. It's very clear, actually, that timing is important for not just for soluble TREM2, but probably for other myeloid targeting strategies as well. So I think you're thinking exactly along the right lines. For this treatment specifically, because we're able to do serial treatment so easily because of the catheter, we're just going ahead and giving the simultaneous therapy each time we give it, because if it's better to give it before, after, concurrently, we're trying to hit all our bases, right? But it may be the case, maybe it does make sense to, for instance, give the CARs first and then don't give the Gemmies until a week later, right? And I think that hopefully that'll become apparent as we do these studies.
I think our last question, Kurt.
Do you think your B7-H3 CAR is targeting the antigen-presenting cells in the tumor?
Yeah, that's a great point, because you're absolutely correct. B7H3 can and is expressed by some antigen-presenting cells, myeloid cells, so that's entirely possible. I think that's entirely possible, and something that we'll look for once we get down the road to doing the immunofluorescence and then flow cytometry studies on the tumors to see if we're clearing those cells as well. Which could be, potentially could be beneficial. Some of those cells are likely to be actually suppressive, not actually activating APCs. So.
Cool. Thank you.
All right, thank you.